Hi students, welcome back. This is part 2 of the integumentary system lecture, and we left off last time learning about the basics of the integument, that the integumentary system is made of two main regions, the epidermis and the dermis. We learned about the 5 distinct layers of the epidermis, and we learned about how we get the different colors or the different pigmentation of the dermis. Now we're going to move on to how the epidermis is attached to the dermis, then move on to details about the dermis, and then move on to some of the accessory structures, and then we'll be finished with the integumentary system. So just as a reminder, and you can see it's labeled here in the photo for you, but the layer that they're kind of pulling away here is the epidermis. So this is where you would find those 5 distinct layers, such as, for instance, this would all be stratum corneum. That's one of the layers, right? And so from here to here is the epidermis. What you find underneath that is the dermis. So from here downwards, it would extend a little bit further than that, but the picture cuts off there. That's the dermis. And both of those together, the epidermis and the dermis, make what we call the cutaneous membrane. So epidermis plus dermis is the cutaneous membrane part of the integument. So if we look real quick at how the epidermis is attached to the dermis, you don't see that the epidermis has a flat, a flat surface. Rather, it has these little ridges right here. These almost like projections. Those are called epidermal ridges. And then if you look at the dermis has these little hills and valleys. And those are called dermal papillae. And so the epidermal ridges, as you can see over here in the middle of the photo, fit right into those dermal papillae. Imagine interlocking your fingers, so sliding the fingers of your right hand between the fingers of your left hand, and then kind of giving that a squeeze. That's a nice, tight connection between your two hands. And that's how these epidermal ridges fit right in those dermal papillae in order to make a nice, tight connection between the epidermis and the dermis. This connection can be broken. So if you are sliding the skin back and forth, let's say you got yourself a new pair of shoes and they don't fit quite right yet, and the shoe is rubbing on the skin of your ankle, it's rubbing that skin back and forth like this, that can dislodge the epidermis from that underlying dermis. And then what you got is inflammation and fluid that builds up between the epidermis and dermis. And that fluid ends up pushing up through the dermis, and what you see on the surface is a blister. So that's essentially what a blister is. So that's kind of a segue into learning now about the dermis. So in this picture, again, actually from here to here is the dermis. And that's going to be made up of 2 different types of connective tissue that we've learned about before. So we're going to pop back here to this, this image. And in the image, you see your authors have shown you in brackets that this is the papillary layer. So all of this would be the papillary layer. And the blood vessels that you find here are part of what we call that papillary plexus. These are the blood vessels that are going to be delivering nutrients to the epidermis. They don't penetrate the epidermis. They're only found in the papillary layer, but it's these blood vessels that that are going to carry nutrients as close as they can to the epidermis. And here are my thoughts on blood vessels. Obviously, we don't learn about blood vessels until later on in the semester, but this is kind of a good introduction for you. Blood vessels are like roadways. Roadways carry us from main locations to very specific locations, and from very specific locations to main locations. So imagine, for instance, let me see if there's a good place to draw this. All right, so let's think of your house. Where you live is right here. See, worst artist ever. But there's your house. And here's Miracosta College. Right down here, main location. So you leave Miracosta College and you're going to get on some major roadway. Maybe it's the 78. Maybe it is the 5. Whatever it is, you're going to get on that main roadway. And then eventually, as you get closer to your destination, you're going to get off the main roadway and you're going to go down a smaller roadway, maybe for some period of time. Then you're going to get closer to your house and you turn off on a side street that's smaller. And then you get really close to your house, and then ultimately you end up in your neighborhood. And what happens when you pull in your driveway or you pull up on the street in front of your house is you get out of your car. And then you go into your house, right? This is the way that nutrients and oxygen and other gases make their way to the epidermis. They're going to be traveling, those nutrients and gases are going to be traveling in those blood vessels and ultimately end up at their destination. And then those nutrients and gases are going to diffuse out and then diffuse into this the epidermis. So that papillary plexus plays a major role in not only nourishing the surrounding connective tissue here, but also penetrating and nourishing the epidermis, that epithelial tissue that is avascular. Okay, so that's a little bit of a sidetrack, but I wanted to explain to you the importance of that papillary plexus. And then the other layer, the deepest layer of the dermis is, let me see what color I should use. I'll use yellow. The deepest layer of the dermis is called the reticular layer. And that makes up the remaining 80% of the dermis. And that reticular layer is made of dense, irregular connective tissue. And it has its own network of blood vessels as well. So let's go explain them to you. So let me erase some of this so it's not too busy for you. Okay, sensory receptors embedded in the dermis include what we call lamellated corpuscles. You might see this written as a different term. You might see this called a Pacinian corpuscle. Either term is okay with me. Just so you know that there are two possible terms, depending on where you're reading. And there are also what we call, the book's calling this a tactile corpuscle. You might see that, but in my next slide, we call it a Meissner's corpuscle. And tactile just means touch, but really a Meissner's corpuscle and a lamellated corpuscle are both stimulated by touch. They're both generally categorized as being tactile receptors. So that's why I think using the term Pacinian and Meissner's is a little bit easier, because in your book, they've just referred to that receptor as by its general term, the tactile corpuscle. But to differentiate it, we would call it a Meissner's corpuscle. You don't see free nerve endings in this picture, but I will show you them in another picture. So those are the two sensory receptors. Let's have a look. And we've lost some formatting here. I'm not sure why that happens. But the superficial most layer is called the papillary layer. That's the superficial 20% of the dermis, or the 20%, the 20% of the dermis that's closest to the surface. And that's made of areolar connective tissue. It's got blood vessels in it, and those blood vessels create this network that we call the papillary plexus. Plexus just means network. So you've got this network of blood vessels that are weaving themselves through this areolar connective tissue. To this slide. So that reticular layer is the deep 80% of the dermis. We say it's deep to the papillary layer, and it's composed of dense irregular connective tissue. In fact, when you were learning about dense irregular connective tissue in the connective tissue unit, you were looking at the reticular layer from the skin. So you've seen this before underneath the microscope. That the network of blood vessels there is called the cutaneous plexus. So that would be like the major roadways that I was just explaining. So those are larger blood vessels. So those larger blood vessels come together to create the cutaneous plexus. Point them out before or tack. We can have a look at this underneath the microscope, and it's pretty clear once you've kind of been told what's what. This is the papillary layer, and that would be made of areolar connective tissue. And then down here you should recognize what type of tissue that is. The name of the layer is the reticular layer, and that's made of dense irregular connective tissue. I'll just abbreviate it. And that papillary layer is made of areolar connective tissue. And even if you don't recognize the tissues immediately, you can recognize that there's a difference. Visually, there's a difference between what you're seeing here and the tissue type that you're seeing here. So those are the two main layers that make up the dermis. Let me go back to one of these and receptors. So that's all I'll mention about the dermis. But now I'd like to move on to the sensory receptors that are accessory structures of the integumentary system. And most of them we find in the dermis. So find in the dermis. So I'm going to move to another picture, and I'm going to say all of this. So I won't read this to you. You can use this slide just as a means of study. Let's move to the picture, and I'll explain these structures once we are looking at the picture. Okay, so on the top row, you can see what these structures look like underneath the microscope. And on the bottom, then you see drawings of each one of these structures. A Meissner's corpuscle is found right on the boundary between the epidermis and the dermis. So let me use a yellow pen so this stands out. If this is epidermis, and then all of this from here to here is dermis, what you can see here is an anomaly in the tissue. That would be a Meissner's corpuscle. And a Meissner's corpuscle is a really highly modified structure where in the center you have this nerve in yellow, and then surrounding that nerve you have a bunch of connective tissue. I'll use red. So all of this would be connective tissue that we've already learned about, right? What Meissner's corpuscles do is they detect very light touch. And the way they do that is if forces are applied to the surrounding connective tissue, like if somebody were to hold your hand, for instance, there's a little bit of pressure put on the surface of the skin, the connective tissue compresses and stimulates the nerve on the inside. And that nerve then sends a signal to the brain that somebody is holding your hand. And the Meissner's corpuscles are going to be found in hairless areas like your fingertips and your palms and your lips and the bottoms of your feet, where they're those areas are very sensitive. They're very sensitive to very light touch. And that's essentially what Meissner's corpuscles are meant to do because they're so superficial. They're right on the border between the epidermis right here and the dermis that you see below. So it's not going to take much force put on the epidermis to push down on that Meissner's corpuscle and stimulate it. Now, kind of the opposite, I shouldn't say the opposite, but similar to but different than a Meissner's corpuscle is this, a Pacinian corpuscle. And they're found deeper in the dermis. So it's going to take more pressure to stimulate those structures. The construction is very similar, where you have a nerve here in the center, and then all of these swirls that you see surrounding the nerve are connective tissue. So when force is put on the connective tissue, that's going to compress or stimulate the nerve fiber here, and then a signal gets sent back to the brain about deep pressure or deep movements or feeling deeper in the dermis. So you might stimulate a Pacinian corpuscle by grabbing your left arm with your right hand. Go ahead and try that and squeeze really hard. And you'll feel or perceive that a feeling of squeezing your arm because of those Pacinian corpuscles. So the reason for having these structures in the body is to let the brain know what's going on the surface or just below the surface of the skin. The importance of knowing that is, do you need to make some adjustment? Is that pressure or is that force going to cause tissue damage? Or is there something crawling on my arm that shouldn't be crawling on my arm? An ant, a mosquito, a fly, a centipede, or whatever the case may be. So these sensory receptors are constantly sending information back to the brain about what's happening on the surface of the skin, so your brain can make a decision about what to do about that stimulus, how to react to that stimulus. Without those receptors, essentially the skin would become almost numb. You would lack the perception of those types of feelings without those receptors. Now, these aren't the only types of receptors you find in the skin. I'm just giving you a couple of examples. If we move to this picture right here, which is from a different textbook, you can see the Meissner's corpuscles here. So see how close they are to the epidermis? So those are stimulated by really light touch. And then you can see the lamellated corpuscles, and you see how much deeper they are in the dermis. So they're going to be stimulated by deep touch. And then the last sensory receptor I'd like to explain to you are free nerve endings. So these structures. You see how they extend into the dermis? They are not surrounded by connective tissue like the other two receptors are. They're surrounded by epithelial cells. So this is why they get their name free nerve endings. They're free of surrounding connective tissue. So those free nerve endings are going to be very sensitive to a number of stimuli. Could be changes in temperature, could be a caustic substance on the surface of the skin. It could be that the skin is wet or dry, a variety of different stimuli. So those are kind of the oddballs. Those free nerve endings are not in the dermis, whereas the Meissner's corpuscle and the Pacinian corpuscles are embedded in the dermis. So those are two accessory structures of the integumentary system. One just kind of fun thing is lines of flexure or flexure lines. These are what you might have read if you were to go to a palm reader. So if you look at your palm, you can see that you have some pretty clear lines at areas of articulation or at your joints, right? And so what gives you these lines of flexure is where the dermis, the underlying dermis, attaches tightly to structures around the joint. And because joints are areas of movement, you have movement of that connective tissue there as well, and that's what gives you those lines. They're caused by continuous, I shouldn't say continuous flexion, but flexion. You see them when you flex in those areas. You've got them on the wrists, the fingers, the toes, the soles of your feet. You've got them at the backs of your legs, the antecubital region of the arm, of the elbow, right? So you see them in a variety of places where flexion is occurring. Okay, and then another accessory structure of the skin, these are hairs. So there's a lot of really neat information to learn about how hairs grow, and we don't really have time in our class to discuss it, but if you look at this picture right here, all of this is epidermis, right? And you see that the purple area of the epidermis kind of comes down like this. It invaginates, comes down, and then wraps back up around the side. So that invagination, or that dipping down of the epidermis into the dermis, creates what we call the hair follicle, the structure or the organ that creates or produces hair. And hair, as you know, is made up of epithelial cells, stratified squamous. It's just that when these squamous cells are growing, they're growing towards one another and being compressed into this structure that is a hair, rather than the epithelial cells growing from the deep layers out. So those cells grow together and get compressed into this singular cylindrical structure that we know as the hair. The shaft of the hair is the exposed part that's poking out of the skin. The bottommost, or the deepest part, is a structure that we call the hair bulb, and you see some blood vessels there. So these blood vessels are delivering nutrients to the hair bulb so that the tissue can continue to grow and create hair. And then if we move up, you see this structure right here. And that's called an erector pili muscle. And it's made of smooth muscle, which we haven't covered yet. It's made of smooth muscle, and smooth muscle is involuntary, meaning you don't have control over it like you do many of your other muscles. And so this is attached directly to the hair follicle. See that? And when this muscle contracts, it makes the hairs on the surface of your skin stand up. And there are two reasons for that. One, when your hair stands up, you are able to trap, I should use a different color, pardon me. You're able to trap a very thin layer of air between the external environment and your skin, and your body heat is going to heat up that layer of air. So here's body heat coming out, and that's going to warm this air in this space just a little bit to keep you warm. So it doesn't really play a huge factor for us as naked Homo sapiens, but in our early ancestors who were much hairier, imagine how dense the hairs would be. So if you have all of those hairs standing up, then that's going to create a nice thick layer of air between the body and the external environment that is warmed by the body and then helps to maintain body temperature. Another reason that we may have evolved these hairs is to make ourselves, or sorry, not the hairs, but the erector pili muscles. Another reason why our hair stands on end is to indicate either excitement or being afraid or fear. So imagine our early ancestors that are much hairier than us. If when they were scared or excited, all of the hairs stood up at once, then that would be an indication to another member of their species that, hey, I'm excited, there's something going on here, come look. Or it could indicate to a predator, hey, look how big I am, look at how big look at all my hair standing up. Doesn't that make me look bigger? You probably don't want to eat me. So in that respect, it's a protective mechanism, right, or a deterrent. But that's why we think we still have these erector pili muscles to either communicate a feeling or a state of being, and to make ourselves look bigger. If you've got a cat at home, when your cats get scared, the hair on the back of their back stands up straight, and it makes them look a little bit bigger, like, hey, don't eat me, or you don't want to mess with me, kind of thing. Okay, so that was the end of the dermis. We learned about what the dermis is made of and the accessory structures that we find in the dermis. I'm really just going to skate right past what we call the hypodermis or the superficial fascia, because we've covered this before, but let me outline it for you. This is going to be the superficial fascia. It's made mostly of adipose tissue, and what it's going to do is it's going to connect the cutaneous membrane, which is all of this, and its accessory structures for that matter, to underlying muscle. So imagine underneath here would be muscles, so that superficial fascia is going to anchor those two structures together, the cutaneous membrane and muscles. It also is very well vascularized, keep that in mind, right? So you've got those blood vessels there. To gland. This picture right here, here's a sebaceous gland. You only find them attached to hair follicle. And then we're going to move on to, now I'm not sure why these slides got mixed up. They should have been the glands slides. This information should have come right after hair follicles. So I lost some formatting. So we're going to cover glands. They are also accessory structures in the dermis, not in the hypodermis. So accessory structures of the dermis include sensory receptors, hair follicles, and glands. So even though these slides are not going in order anymore, you still have that organization. So when we were learning about epithelial tissue, we learned about two types of glands. Remember, there are exocrine glands and endocrine glands. The endocrine glands are ductless, no duct, and they secrete hormones. So we're not talking about those. We're talking about exocrine glands. They secrete a product onto the surface of something. So it could be the surface of the skin, the inner surface of the stomach, the inner surface of the trachea. So these exocrine glands that we're going to talk about are exocrine glands that are secreting something onto the surface of the skin. So there are sweat glands, and there are two main types of sweat glands. So first one, merocrine sweat glands. So these are sweat glands that you're probably most familiar with that secrete a salty sweat for reducing body temperature or thermoregulation. they're not really serving an excretory function. We're not like getting rid of excess salt by these glands, even though the fluid is a little salty. It really doesn't perform any excretory function. It's really just used for thermal regulation, for bringing our temperature down. And these can be found universally all over the body. They use the Exocrine method of secretion. So go back to your epithelial tissue lecture if you've forgotten what Exocrine secretion is. The other type are kind of fun. I love talking about these. These are what we call apocrine sweat glands. And instead of secreting a watery, salty fluid, they secrete a fatty and proteinaceous fluid, which seems counterintuitive because fat is a very good source of energy and we need proteins, right? So we don't normally like to get rid of those. If we can hang on to those things and conserve them, that's great. So we think that the reason that we have these apocrine glands is for communication. Think about a couple 100,000 years ago. There was no language. We were completely covered in hair, and we needed to communicate with other members of our species. What types of things would you like to communicate? You might want to communicate your sex. You might want to communicate your age. Are you of reproductive age or not? You might want to communicate if you're scared, fear. You might want to communicate whether you're family or not. So prior to having language, we think that these fatty proteinaceous products that are secreted from apocrine sweat glands were really important in communication. And where we find these sweat glands are in the axillary and inguinal regions, so in the armpit and the groin. And these tend to be the areas where body odor occurs most frequently. And the reason for that is bacteria really like to munch on these fats and proteins that are secreted. Now, the bacteria themselves don't create an odor, but their metabolites do. What they metabolize or their waste products from digesting fat and protein are what cause that characteristic body odor smell. One last thing I want to mention is that these apocrine sweat glands, you would think that their method of secretion is apocrine method, but in fact it's the Exocrine method. When these sweat glands were first described, it was thought that they used the Exocrine method. Turns out they don't. They use the apocrine method, but scientists didn't want to change the names. So that's that. Back to. And then lastly, there are sebaceous glands. And sebaceous glands are only glands that are attached to hair follicles. And these sebaceous glands secrete this kind of lubricating and moisturizing oil onto the hair itself, but more importantly, onto the skin. And that helps to keep the skin moist, keeps it healthy. We think that this sebum, this fluid, also has some antibiotic properties to help us keep the bacteria that live on our skin in check. And so if you haven't washed your hair for a few days, or you haven't taken a shower for a few days, when you kind of get that greasy feeling, what's collecting on your skin is sebum. So you've got these sebaceous glands everywhere that you have hair or hair follicles. So where you don't have hair or hair follicles are the palms of the hands and soles of the feet. So you wouldn't see sebaceous glands there, only associated with hair. They use the holocrine method of secretion, and when they get clogged, this is what causes acne. So a blackhead is clogged with sebum, just sebum, and when that sebum comes in contact with air, it turns black. So when that sebum is oxidized, it turns black, and so that's what makes blackheads appear black. Whereas a whitehead is different. bacteria make their way into the follicle, white blood cells rush to the follicle and come in to kill the bacteria, and that's what produces that pus. So really that pus is white blood cells at the site of infection of the hair follicle. And then we're going to move on to something a little bit more clinical related, and that's severe burns and the skin. So there's the, and I think this has been modified a little bit in the last few years, but we call this the rule of nines. And the rule of nines means that we break the body up into areas of 9% of its total surface area. So the anterior thorax, the skin on the anterior thorax, makes up about 9% of your overall surface area of your skin. The anterior arm, 4.5%, and 4.5% on the other side makes up another 9%, and so on. So this is how we classify burns, and we classify them percent surface area that's been damaged. But we also classify them based on degree. So you might have heard somebody or heard or read about somebody with, for instance, 3rd degree burns over 30% of their body. The 3rd degree refers to the depth of the burn, and the percent that they're indicating refers to the amount of surface area of skin that has been burned. I'll get to the degree part in a second. So even shallow burns, if they cover large enough areas of the body, can be fatal. And the reason for this is if you remove the epidermis, for instance, which would be a really superficial burn, would be a 1st degree burn, you are susceptible now to losing electrolytes from the skin, sodium, potassium, magnesium. All of those electrolytes are really important for cellular activities. But you also have removed your outer coat. So you begin to lose the ability to thermoregulate. You get too much water evaporating from the skin, and your body begins to cool. And at a time when you should be putting all your energy towards healing, your body's now having to put energy towards just maintaining 98.6. So this inability to thermoregulate is a complication of a burn. Loss of electrolytes is a major complication of burn. If I had to prioritize these, excuse me, the oozing of the skin would be the worst complication of a burn. Then inability to thermoregulate, and then infection. Yes, the skin plays a really important role in keeping out pathogens, but this would be like a tertiary concern. We have a clinician or a doctor or a nurse would have to get control of the oozing of the skin, and then keeping the patient warm, and then worrying about infection, because numbers 1 and number 2 can cause fatality. So can number 3, but number 1 and number 2 would be the areas of first concern. Okay, so skin has these slides are... Okay, so now we've talked about damage. Let's talk about how skin repairs itself. We know that we've all had cuts and bumps and bruises, etc. So we know that the skin repairs. Remember that epithelial tissue, all of it has a high mitotic rate, meaning those cells grow and divide pretty quickly. And they'll regrow quickly if the cells are injured. But you must have that deepest layer of the epidermis healthy in order for the skin to regrow. So the stratum germinativa, remember that is also called the stratum basal. That's the deepest layer of the epidermis. That's the mitotically active layer, the cells that are growing and dividing. So those need to be present in order for the skin to regrow and seal the injury. If no stratum germinativum is present, doctors can take stratum germinativum and skin from elsewhere to do what we call a skin graft. The area also needs to have a good blood supply. That skin's not going to grow without a good blood supply. So if the blood supply is still intact, the cutaneous plexus and the papillary plexus of the dermis, if those are still intact, fantastic, then repair can occur. And then even the dermal damage can be repaired. So all of the above, we were talking about regrowing the epidermis. The dermis can regrow too. And what will happen, it will just add more collagen. So remember that dermis is made of connective tissue, and it will replace or it will regrow collagen fibers to kind of seal up the injury. And that's what results in what we see as a scar. Out of order. I really apologize. Let's talk about the degree of burn. So we already know that we classify burns based on the percent surface area that's been damaged, but we also classify burns based on how deep the burn goes. So a first-degree burn is pretty minor, where you just have the first superficial epidermal cells that have been damaged or killed. You'll probably get a little bit of inflammation and tenderness, but the hair follicles are unaffected. The sensory receptors are unaffected. All those structures in the dermis are unaffected because you've only damaged the epidermal cells. So this would be like, I don't know, touching a hot stove. A second-degree burn is where you have both the superficial and deep epidermal layers have been damaged or killed. You might have injury all the way down to the dermis, but not all the way through the reticular layer. And even in this case, most accessory structures remain intact. So most sweat glands, most sensory receptors, most hair follicles, they're all going to remain intact. But here now you're going to get blistering and pain. So this might be like having a bad sunburn, and you get a blister from a sunburn and pain from a sunburn. And then third-degree burns, you have all the epidermal and dermal cells killed. So you could have damage down to the subcutaneous layer. Subcutaneous layer is another layer for hypodermis, which is also another name for superficial fascia. The reason I'm using the term subcutaneous right here is because that's a clinical term. So in a third-degree burn, you get all the epidermis, all the dermis, damage all the way down to the hypodermis, and the skin is going to appear charred or burnt. The nerve endings, or the sensory receptors, they're destroyed. So initially, these burns are not painful. And it seems kind of counterintuitive, but because there are no sensory receptors, the sensory receptors have been damaged or killed, there isn't any immediate pain. The pain comes from when healing begins, when the epidermis and dermis are still coming back and those sensory receptors are really close to the surface. And when those sensory receptors are growing back, that's when they're very, very sensitive. Okay. So this last slide I would like you to work on your own, and should have a title. I would like you guys to know some of the things that happen to the skin as we age. What happens to melanocytes? What happens to the hair follicles? What about skin repair? Are we better or worse at repairing skin as we age? So all of these points here, I'd like you to be familiar with. Okay, you might want to make yourself a chart. You can do this in any number of ways. Okay, that's a little activity for you to do. All right, that includes part 2 of the integumentary system lecture. See you next time.